I'm going to tell you about fungi that live inside of plants and how we're studying all aspects of their biology and evolution. So just like we now recognize how important symbionts are for our own human health, I think there's increasing recognition of how important microbial symbionts are for plant health and productivity. And so in particular, symbionts of plants are known to basically allow plants to deal with abiotic stress. And so here I have kind of an extreme example of that picture of the whitebark pine in Crater Lake National Park. And so this particular species of tree is known to associate with over 30 different mycorrhizal fungi that allow it to live in these extreme environments. And so in particular for this species, and for a lot of other plants, so these mycorrhizal associates provide plants with resistance to desiccation, to nutrient stress, and protection against herbivory and pathogens. And so given the importance of these fungal symbionts, it's not surprising that plants and fungi have co-evolved with each other over the last approximately 500 million years. So here is a figure that I modified from Martin et al., basically just showing how root-associated fungi have associated with plants. So on the left, you can see that the earliest fossils of land plants are shown to have mycorrhizal-like associates. We have the early relatives of conifers that are associated with arbuscular mycorrhizal fungi. And then the first Pinaceae species likely co-evolved with the earliest ancestors of the ectomycorrhizal fungi. And so as you can see, these sorts of root symbionts are extremely important for plants' ecology and evolution. But it's not only fungi in the roots that are important for plants. And so fungi can also inhabit the above ground tissue, the photosynthetic tissue of the leaves, and that those fungi can provide plants with resistance to stress. And so here I've got an example of a tropical panic grass. And so this panic grass can live in geothermal soils that reach temperatures of over 45 degrees Celsius. And the ability to do so has been attributed to a fungus that lives inside of the leaf tissue, and then that fungus in symbiosis with another viral symbiont then allow this plant to live in this extreme environment. In addition, above ground foliar fungi can help protect plants against pathogens. And so in this elegant experiment by Arnold et al., Theobroma cacao leaves were inoculated with foliar fungi that can protect it against a pathogen. So on the left is a leaf that shows it was inoculated with the symbiont and then a pathogen, and there's no symptoms of disease. On the right hand, you see that it was only inoculated with the pathogen, and you can see the diseased leaf tissue. And so these fungi that live asymptomatically within their host species are grouped together functionally as endophytic fungi. And so again, they're asymptomatic. You cannot see evidence of their infection with your naked eye. And these are healthy plant tissues. There's no obvious signs of those infections. So typically in woody plants, endophytes are found at least in the leaf tissue, in the intercellular spaces. They're highly diverse. So in the tropics, for example, a single plant leaf can harbor dozens to hundreds of species of fungi. They're horizontally transmitted. So spores and hyphal fragments are transmitted by wind and rain to new species. And one thing that I want to mention is that they've been primarily studied early on with culture-based techniques. And so here's a beautiful picture from Arnold showing the morphological diversity of these endophytes. And so today I'm going to focus on talking about endophytes that live inside of the above ground photosynthetic tissue. Even though we know now that there's endophytes that can associate with bark in the vascular tissue and also endophytes in roots. And so one of the most amazing things about endophytes that live in photosynthetic tissue is how diverse they are. So this hyperdiversity of follar endophytes was first documented by Arnold and Luzzoni, where they did a latitudinal transect, and they demonstrated that the highest endophyte diversity is found in plants in the tropics, and then similar to macroscopic organisms, that the diversity decreases as you head toward the poles. But one thing that they noted was that actually the phylogenetic diversity of endophytes was greatest in boreal forests. And so here they measured phylogenetic diversity. So the top right hand is the pie chart represents the number of different classes of Ascomycota, and you can see that there's more classes of Ascomycota found in endophytes of boreal forests than there are in the bottom right hand, where you show that there's really only 2 dominant classes in tropical forests. And so even though species diversity declines with latitude, phylogenetic diversity increases. And so let me just say that this study was, again, based on culture-based work, but the same pattern was found with 454 data with this meta-analysis where they basically showed that the green dots are the species richness from living plant tissue, and then again, that diversity decreases as you go towards the poles. But one thing I want to point out is that in these studies, the plants that they focused on were angiosperms and conifers. And so one thing that when I first got working with endophytes, I was interested in what happens if you compare the richness in different biomes across all different types of land plants. And so I sampled angiosperms, conifers, ferns, lycophytes, and mosses in 5 sites across North America, 3 temperate sites, and then 2 in Alaska. And on the y-axis, I'm just showing the diversity of endophytes per species per site. And what we found in that study was actually there's no significant difference in richness for endophytes in these different biomes. And so in particular, what we found was that there was an incredibly high diversity of endophytes that associate with lycophytes and mosses, especially in the northern biome. And I'm not showing it today, or not going to talk about it today, but there's also an incredible amount of endophyte diversity found within lichens. And so probably not many of you think about lichens, but lichens are a symbiotic association between a fungal mycobiont, which provides the house, and then you have a photosynthetic, usually a green alga photobiont that then provides the food. And so in addition to that main algal or cyanobacterial photobiont and the mycobiont, you also have all these other fungi that live inside of these lichens as endophytes, and they're more similar to endophytic fungi of plants than they are to other lichen-forming fungi. And so one aspect that I've been trying to focus on then is what is the evolutionary context of the endophyte symbiosis when you look across all different lineages of plants and green algae and lichen thalli. And then also, so on the left is a phylogenetic tree of lichen-forming fungi and the Lichenoromycetes. And so we've sampled endophytes in all of the clades that are highlighted in red, and then again, all of the lineages of land plants that are highlighted in green. And so this gives us an understanding of how are endophytes associating with all these different lineages of photosynthetic organisms. And so in this evolutionary context, then, I'm really interested in what are the interplay between ecology and evolution? So where are endophytes found, both across different hosts and across different geographic locations? And then importantly, who are they? I think Bart gave a really important example of why the tree of life is important. So most of what we can culture from leaves of all different plants and from lichen thalli, most of these represent novel species. And so when we put these in a tree of life, we often find there's no close relative. And so one thing that's challenging about this is that basically when you use like short read technology, it's really difficult to answer who. And so this is where cultures and then now PacBio sequencing would be really important. And then also I'm interested in obviously in what they're doing. And so I don't have time today to talk about all of the PacBio genomes that we're sequencing to try and answer what they're doing. But today I'm going to focus on kind of a study that we just did where we're looking at where fungi are found in plants and then who might they be and how they evolved. And so I'm going to talk today about a postdoctoral project I did on this dimensions of boreal endophyte diversity. This was an NSF-funded project, and this was in collaboration with Betsy Arnold at University of Arizona and Francois Luzzoni and Jolanta Midolakowska at Duke University. And so we sampled endophytes in 60 different host species per site across these sites that are marked in the boreal biome with stars. And so in total, we had almost 500 different host collections. And so one of the first things we wanted to do, as I mentioned, that all the seminal studies on endophytes were first done with culturing, and then of course with next-gen sequencing, culturing was kind of put to the side, but what we wanted to address was actually what are the differences and inferences between using culture-based versus culture-free approaches. So for each of these species, we collected the tissue, we washed it, we surface sterilized it with sequential washes and ethanol and bleach, and then we cut it into small 2 millimeter square pieces. And 96 of those tissue pieces were randomly selected for culturing and then Sanger sequencing. And then the other 96 were put into CTAB for DNA extraction for Illumina next-gen sequencing. And so with this, we could directly compare the same amount of tissue, what is the richness of endophytes we get. And I should mention, you're all thinking, why don't you just do whole genome shotgun? The problem is that within these leaves, even though you can have a lot of high biodiversity, actually the biomass of fungi is really small. So maybe 1% or less than 1% of the DNA of any leaf would be fungal. And so you would primarily just be resequencing plant genomes. Not that there's anything wrong with that, but so again then for the culturing, so we were sequencing kind of the fungal barcode locus. So here we have the conserved regions of the ribosomal operon shown in the brown, and then the more hypervariable ITS1 and ITS2 regions shown in the dark brown. And so with the cultures, we can sequence from ITS1 through a portion of the large subunit, but with the Illumina next-gen sequencing, we're limited to only sequencing about a 300 base pair fragment of that hypervariable region. And so I'll say the hypervariable regions are really useful for community analyses, understanding what is the species overlap, but they're so hypervariable that they can't be aligned, and so we can't use them for phylogenetic analyses. And so in this study, then we had almost 11,000 fungi that were cultured from around boreal forests around the world, and these were all sequenced with Sanger for the ITS partial large subunit nuclear ribosomal DNA. And then paired with that, we had over 20 million MySeq ITS2 sequences. And the power of this is that if we want to understand how endophytes are distributed across different plant species and across space, large geographic scales, the problem is usually if you go across large geographic scales, climate differs, therefore you get different plants. And so the advantage of doing this in the boreal biome is that we can span different geographic scales all the way to the global scale while keeping while sampling the same plant and lichen species, or at least the same genera. So first, I just wanted to say a little bit about the complementarity of culturing and culture-free approaches. And so in total, we had about 10 million of these ITS2 sequences from NGS with the cultures, and that yielded about 6,500 operational taxonomic units, or a proxy for species. And kind of not surprisingly, if we look at a Venn diagram of the overlap, we see that we had 43 OTU that were found only with culturing, and then 352 OTU that were found with both methods, and then obviously the majority of the richness of fungi was found using culture-free methods. So in general, we had about 15-fold difference between culture and culture-free methods. And if we look at the taxonomy differences, we find that the light brown here, or well, kind of weird brown color, is a Ascomycota. So both culture and culture-free methods were dominated by Ascomycota. But we see on the right, you had the NGS sequences, and we have more Basidiomycota, and then a lot more unknown fungi. And that could be because we're capturing novel things. It also could be that we have such short sequences that they're really hard to classify. So we recover the same groups of Ascomycota, but we see this large difference in richness. But one thing that I was interested in looking at was what's the difference in richness per species between the two methods. And so here's a regression analysis that shows on the y-axis the number of OTU per host with Sanger sequencing of cultures, and then the x-axis is for next-gen sequencing. And we can see there's a pretty strong correlation between richness for these two methods. And then if you actually subsample the next-gen data down to the same sample size as the Sanger data, we see a very strong correlation. So this suggests that we don't have any strong inherent bias, at least for Ascomycota. It's more of a effort-based problem with what we're, how we can discover the novel biodiversity of these fungi. And so we're interested in how these fungi are distributed. If you walk into a forest and you see all these different types of plants and lichens, how are the endophytes in those organisms distributed. And so to look at that, here's a network diagram. I'll kind of walk you through it. The white circles show the host individuals. The colored circles are individual OTU, and they're structured by the number of different host lineage, or colored by the number of different host lineages that they're found in. So the red are found in a single host lineage. So for example, on the far left, those are all the OTU that are only found in lichens. And then all the way to blue, where you find those are the OTU that are found, and this is in a single community, so for example, in Russia. And so overall, we found that 64% of the OTU in a single site were then associated with only one plant lineage. And so there's this high specificity of endophytes for their hosts. But one thing that we're interested in, we know that locally, they all have the same environment. What we wanted to know is then if you spread across this geographic scale, are you still going to see the same pattern? And so one thing we expected was that with increasing geographic distance, we would see increasing differences in the endophyte communities. And so the top left there is a distance decay plot, but we don't observe any distance decay actually in these communities. And then the bottom is a Mantell correlelogram looking for spatial autocorrelation. The dark circle basically shows that we found significant spatial autocorrelation only for species that were in the same site. So we don't see any strong effect of space. And if we look using a non-metric multidimensional scaling plot, where so each of these points represents the endophyte community from that species, they're color-coded by their host, and then shape is by site. But you can see here that there's, even across 9,000 kilometers distant, that communities are structured more strongly by what host they're in than by where they are geographically. And that's not to say that there's not some geographical component, but by far the strongest impact is what type of host plant you're in. But that's at a community level, and so does this mean then that mosses in Alaska have exactly the same community as mosses in Sweden? Well, not necessarily. If we look at the OTU, how different OTUs are associated with different plants globally. So here's a network diagram that's showing globally how are these endophytes distributed. You see a lot more OTU are actually shared among the different lineages. But again, you still see the red, we joke and call these the turkey plots, that the red are still all the ones that are kind of host specific. So globally, on average, there's still almost a quarter of the endophytes that are only found within a single host lineage. But because you have to think, these are symbionts of plants. And so to be successful, they not only have to disperse, but then they also have to find a compatible host. And so we might expect then that the geographic breadth of an individual species would be greater if it were able to interact with more different types of plants. So therefore, we tested the hypothesis that like host generalists would be more geographically widespread. And so here I'm just showing the colored bars represent the number of different host lineages an OTU associates with, and then this is within a single site. So again, you can see that over half of the OTU in a single site are associated with a single lineage. Well, if we look at that then across all the sites, okay, if you follow it to the right, so you have, these are OTU that are found in all 7 sites, and you see again, most of those then are associated with more host lineages, so with the blue. So it looks like geographically widespread OTU are more likely to be host generalists, and that's not too surprising. It makes sense. But one thing that we're interested in then is how do, if we look at this in an evolutionary context, can we see how these sorts of relationships evolve in different lineages in different places? And so here we took the 10,000 or so Sanger sequences that we had. As I mentioned, that they have the more conserved regions that we can place into this phylogenetic context. And here is a phylogeny of the Dithydiomycetes. And what you can see on your left, so the inner circle represents the colored by the host associations. The middle is the site, and then the outer is the continent that they're found in. And so on the left, for example, you can see the large clade that's associated with purple. Those are all found in lycophytes. And most of those are found in North America. And so we see this pattern within this phylogenetic context. And then in contrast, on the right side of the phylogeny, you see that there's clades that are more host generalists. But again, we're limited here. We recovered only a fraction of the diversity using culturing, but those are the only ones that we can really put in this robust phylogenetic framework. And so what we're trying to do now is to answer kind of both where and who these are using the PacBio sequencing. And so this has been done for aquatic fungi and for soil fungi, but I don't know that this has been done for plant-associated fungi. And so just to remind you, we have the ribosomal operon here. We could have sequenced just the same 1.2 kb that we did for the cultures, but what I wanted to do is actually sequence the entire length of the subunit, so that's about 5.5 kb. And so we PCR amplified this. I wrote, so this is kind of new study, some unpublished data, where we collected plants, 25 different species of plants and 3 replicates. Again, we surface sterilized the leaves because we're interested in the fungi that are inside, extracted DNA, and then amplified with these fungal specific primers. So we have, again, a mixture of plant and fungal DNA, and we're targeting the fungi with primers. Then, of course, we do the second round of PCR to add the barcodes and the adapters, normalize, pooled an equimolar amount, and then sequenced on a SMART cell with a SQL2 at the Arizona Genomics Institute. And so the raw data, we had a lot of raw data, which was awesome, and over 12.5 gigabytes from the CCS analysis. So it was a beautiful data set. And I wanted to, this sequencing was made possible through a grant from PacBio and the Arizona Genomics Institute. So I'm really pleased that the data look so amazing. So with these data, I found we had over 1 million high quality reads after stringent QC and chimera removal. As I mentioned, we have this large database of culture sequences, and so one thing that was really nice to see is that we had a number of exact matches to things that we have cultured from the same host species. And so we had very high quality data that again have been validated with all the culturing that we've been doing over the last 12 years. Just taking the ITS region and separating out those different loci, we were able to cluster the sequences in over 2,000 different OTU. So I know in the past people have said that you could do PacBio sequencing, but you're not going to recover the same amount of richness that you would with Illumina. Well, here we did Illumina on the same species, and we got as high of richness as we did with the Illumina. Again, we weren't able to multiplex as many, but we recovered a high richness of fungal OTU. And then what we were looking for and what we found was this higher phylogenetic diversity. So with the Illumina sequencing, we're limited to this particular primer that excludes plants to amplify fungi, but we also know that it excludes a lot of fungi, that it's kind of biased towards Ascomycota, and we don't see a lot of other fungi. And so here, the primers that we're using to amplify this full-length ribosomal unit are not as biased. We don't think there's much bias, and we recovered Ascomycota, Basidiomycota, and all these really neat early diverging fungal lineages that haven't really been known to associate as endophytes before. And so I know I went over my time, but I just wanted to say that our kind of next steps here are basically just to do more phylogenetic analyses. This tree is an example of what we could do with TBAS, which Ignacio Carbone has developed at NC State, where we can actually automate putting these analyses into reference tree alignments and understanding where they go in the tree of life. So I just want to thank all of the Boreal Diversity Project. Was a large multi-institutional collaboration. And then again, I wanted to thank the members of my lab and the genomics resources available at Arizona, all of the funding sources, and then thank you for your attention.